Bismillahirrahmanirrahim. Assalamu alaikum students. Today we are going to discuss the development of the respiratory system. So main objective of the today's slide is to describe formation of the lung bud and discuss its clinical correlates. Formation of the lung bud, the respiratory diverticulum appear as an outgrowth from the ventral wall of the foregut in the fourth week of development and opposite the level of the fourth pair of the pharyngeal arches. In this diagram you can see these are the fourth pharyngeal arches, and here the lung bud is developing. And in this diagram you can see this is the foregut and it is the ventral outgrowth from the foregut. So this is the diagram at 25-day gestation showing relationship of respiratory diverticulum to heart, stomach and liver. In this diagram you can see the relationship of the developing lung bud to the heart as well as to the stomach and the area of the developing liver. So what happens next, that around fourth week of the gestation, the respiratory diverticulum, that is lung bud, appear as an outgrowth from ventral wall of the foregut. The retinoic acid is the main factor involved, which is produced by the epithelium of the foregut. Here the foregut epithelium produces retinoic acid. This retinoic acid then upregulates the another factor, which is TBX4, which induces the lung formation from the mesenchyme and in this area. So in early stages, the lung bud is in communication with the foregut. You can see here this is the dorsal part, which is the foregut, and this area is the ventral bud, that is the respiratory diverticulum, and there is a wide communication between two. Later on, the esophageotracheal ridges appear, which starts to meet in the midline, and when they fuses in the midline, they creates a septum and then divides the foregut into dorsal part, that is the esophagus, and the ventral part, that is trachea, and the two lung buds. So the development of lung buds, next the lung buds form the trachea. Its lateral out pocket known as the bronchial buds, and primary bronchi appears in the fifth week of development, and the secondary or the lobar bronchi appear in the sixth week of development. During further development, lung bud expands in the body cavity. In this diagram, you can see this yellow is the foregut tube, and this darker color is tracheal bud or lung bud, which divides into two, the right and the left lung bud. This area is the pleuroperitoneal canals in which this lung bud is growing. This blue color area shows the visceral pleura, which is developing from the mesenchyme of this area, and this area forms the somatic layer of the pleura, and the cavity between these pleural layers will form the definitive cavity of the pleura. So cavity is known as pleuropericardial canal in which this is developing, and mesoderm of the lung form visceral pleura, and space between somatic and visceral pleura form the pleural cavity proper. So epithelium of internal lining of the trachea, bronchi, lung and larynx is of endodermal origin. So you can appreciate this. This is the foregut, and this is the ventral outpocketing, ventral outgrowth, that is the lung bud or diverticulum. So epithelium is also derived from the foregut, which lines the larynx, the trachea, then the bronchial tree and the lungs completely. But the cartilage, the bone, the muscular tissue, the pleura of the lung and the connective tissue, all factors are derived from the mesoderm in the splenic mesoderm of this area. So these are the two different cell lineages from which lung tissue develop. With further development, the successive stages in development of the larynx are that at 4 weeks of development, when this tracheoesophageal septum is dividing the connection between the esophagus and trachea, there is a junction between the tracheoesophageal trachea and esophagus, which is a slit-like junction, that is the laryngotracheal groove. With further development of other cartilages, this laryngotracheal groove develops into a T-shaped manner, and with the development of the laryngeal cartilages, the cricoid and thyroid cartilages, this T-shaped slit then develops into an adult shape opening of the laryngeal inlet. So these are the clinical correlates. The laryngeal atresia is an anomaly which is a rare one which results in obstruction of upper airway, that is the congenital high airway obstruction syndrome. And laryngeal web is another anomaly which is uncommon anomaly, results from the incomplete recanalization of the larynx during 10th week of development. So membranous web forms at the level of the vocal cords, partially obstructing these airway. These are the two anomalies associated with the larynx, and here are the different anomalies which are associated with the tracheoesophageal septum. So if tracheoesophageal septum does not divide itself properly, these anomalies result in which proximal part of the esophagus ends blindly and distal part of the esophagus has a connection with the trachea, which is called the esophageal fistula. This type of anomaly is common, which is approximately 86% common. 86 to 90% is the rate. In this type of anomaly, there is the isolated esophageal atresia. This type of another anomaly in which upper part of the esophagus and lower part of the esophagus both enter into trachea separately. This is again a rarer form of the tracheoesophageal anomalies, only 1% rate of occurrence. And another type of anomaly which is an H-shaped anomaly in which there is an isolated tracheoesophageal fistula, and the rate of occurrence is 4%. This is another rarer type of the anomaly in which proximal part is attached to trachea by means of the fistula, and the distal part of the esophagus ends in a blind tube. 33% of the cases are involved with the vectoral association. So vectoral is the term which is used for vertebral anomalies, anal atresias, cardiac defects, tracheoesophageal fistula defects, esophageal atresia, renal anomalies, and limb defect. The complication of these type of anomalies are the polyhydramnios, as we all know that all of the amniotic fluid is swallowed by the fetus, and when there is a blind ending pouch, or there is atresia, or there are the fistulas, so this leads to in this case the amniotic fluid will not be ingested by the GIT, and it will accumulate in the amniotic cavity and will lead to the polyhydramnios. This is the X-ray picture of the patient with the vectoral, and in this baby is having tracheoesophageal defect, needed to be carefully monitored for the vectoral. This is the barium meal study in which you can see the blind ending proximal part of the esophagus. So these are the all anomalies of the tracheoesophageal septum and the larynx, and here the first lecture we will finish, and we will meet after this inshallah in the next class. Allah Hafiz.